Lovely, lovely to see you on this beautiful Saturday morning here in Philadelphia. Thank you all for being here. We have a session on gene therapy as a segue to the Large Bio presentation on gene therapy that is around the corner. So we want to unpack that some more in detail and actually hear your voices. Your voices are the center of the gene therapy story. So I am here. I am Jenny Downs. I am from Australia and have come here. So that is wonderful to be here. I am working today with Wendy Gold, a lab scientist, very talented lab scientist from Sydney, who will be presenting. I had the great privilege for us to have a statistician here at today's meeting, and that's Peter Jacoby. He will talk because our work needs statistics to bring all of your voices into something that is a pattern and that we can talk with to people who will want to know what you think about gene therapy trials. Plus, we have three clinicians. You know them, Dr. Scott Demarest, Dr. Eric Marsh, and Dr. Bernard Suter. And everyone here on this panel has very close experience at the coalface in their field. So I look forward to their presentations. We will have the presentations. We will have a short 2 or 3, maybe 4-minute break, and then we will join in tables for discussions. And that workshop session will be hosted by Dr. Jessica Keeley, also from Perth. So we're a mix of a US Australian team here to work with a gene therapy discussion with you. And I thank Karen Utley, who said we want this session in our program, and it's really ignited. Here we are. The ignition is in the catchphrase of this conference, and we're hopefully contributing to that. And we have called, I had better put on my slide. We are calling this Hope and Honest Conversations. And if we think about it, everything you do is talking with your family, with your partners, with your clinicians, with anyone who you are thinking about making a decision in relation to your loved one with CDKL5. And that's about hope, your goals and what you're wanting to achieve. It's always about honest conversations. For example, a new epilepsy med, you have to talk about benefits and risks. But here we are changing the focus of a benefit risk discussion to the benefits and risks associated with gene therapy. And this workshop is for everybody. If you are interested in gene therapy or not interested in gene therapy, that is important. We need all voices to understand a community voice. So thank you all for being here. I think it's great. We've followed on from this morning's presentation about the Large Bio trial, which is absolutely imminent. It's round the corner. And it gave me great hope to hear of the Lulu approach, which is sharing protocols with other pharma companies so that the field will be upskilled and gaining in momentum for this community. So thank you to Lulu and the approach, and thank you to all of my speakers here who will follow me now. The second thing is that here is a workshop where we would like you to join us as a part of a research initiative. We will put out information sheets and consent forms to all of the tables. We would like that so that we can then use the information. We'll have workshops, each of the 8 worksheets. All of the tables will have a facilitator to take your notes, and that will start to help us to develop a survey. And then with the survey, we will quantify community perceptions on risk and benefits for gene therapy trials across a range of scenarios. So this is important information. It centers you in the conversation, and your perceptions, your voice informs clinicians. When you talk to each other about gene therapy trials, it informs what pharma does when they're thinking about gene therapy trials, and importantly, it informs the FDA. So that's why we hope to build a narrative that will uplift knowledge and also perceptions, so we know where to go with gene therapy trials. And we thank you again for being here and being a part of this. I will start the presentations and would like to introduce Dr. Wendy Gold from Sydney, Australia, to give you information on gene therapy. Thank you.
Thanks, Jen. Today's session, as Jen said, is so important because we want to hear from you and how you feel about gene therapies and gene therapy trials. But before we start with these important conversations, my task this morning is to give you a little bit of information about gene therapies and how they work from a molecular perspective before we can talk about how they work as a therapy. So I'm going to start at the real basics. I'm going to assume that a lot of you don't know much about gene therapies. I'm going to take you through the molecular pathways of gene therapies so that you can understand how they actually work in the body. I'm going to start off with how genes and the molecules in your body work. And to start off with, I'm starting really, really basic, and that is, what is a gene? Genes in our body form the instructions. The instructions from the genes allow us to create proteins. And if you think about this as a recipe has instructions. If you follow the instructions, then a cake will be made. And like in your body, the genes are the instructions and they produce proteins. Here's a cartoon of what DNA and genes look like. Basically, DNA are two strands, like two ropes that are curled around each other. And the DNA consists of genes, one after the next, after the next. And the genes encode specific instructions which will provide proteins, and the proteins are the molecules that will allow your cell to function normally. And proteins do the work. They help your body to grow, to heal, to function. Now, the next concept I want to introduce is a mutation. It's often also called a variant, so it'll be used interchangeably. A mutation is a change in a gene, so when something has been changed. In a normal gene, the gene will be converted into a protein, and then the protein will allow the cell to work normally. When there is a mutation or a variant, the protein's either not made at all, so there is no protein, or it's not working properly, and then the cell doesn't work properly. Now, if we go back to our cake analogy, if you have a recipe, the recipe has instructions, the gene, and if you follow the instructions, then a cake is made and the normal protein's made. But in the case of a mutation, if there is a mutation in a gene, it's like a typo. So I don't know if you can see the slides, that sugar is spelt incorrectly. So if there is a typo and you don't follow the instructions properly, then the cake's not made. All of you know what CDD is, but what I want to explain to you is how it works, how it manifests from a molecular point of view. It's a very important gene. It's really, really important for the brain. It's important for cells in our brain, which we call neurons, to connect and to communicate with each other. When there is a variant, a mutation in CDD, it weakens these connections between the neurons, so they're not able to communicate properly. And this can lead to the brain cells that can't communicate well, can lead to seizures, and cause neurodevelopmental challenges such as learning, language, movement, and behavior. All right, now I'm going to start producing some concepts about gene therapy. Gene therapy is the introduction of a normal working copy of a gene, and it's necessary to restore production of a working protein. I'm going to introduce some new words, a vector and an AAV. AAV stands for adeno-associated virus. How gene therapy works is we package a gene of interest, it'll be CDKL5, into a vector, into an AAV. This then is introduced into a cell. The gene is expressed, normal protein is made, and cell function is restored. So what is an AAV? AAVs, or adeno-associated viruses, are tiny little viruses. They're so small that you can hardly see them under the microscope. And they get their name, it's a very long name, so we just call them AAVs. They get their name because they are associated with a larger virus called adenovirus, and they need this adenovirus in order to replicate. It contains a structure called, which has a capsid, which is like its outer shell, and it has a genome in the middle of it, and it doesn't have an envelope. And what's important about this is that when we create gene therapies, we gut out the DNA of the virus, and we introduce the gene of interest, in this case it's CDKL5. So we take out the viral DNA, and we can use it to deliver a working copy of CDKL5. Now, these viruses are really versatile and important, and they're non-pathogenic. They have low immunogenicity. They have broad tropism. Here's another word that I want to introduce, tropism. And the reason why it's so important is that there are lots of AAVs. They're naturally occurring, and a lot of us would already be infected by AAVs because they're naturally occurring virus. They have like a GPS system, and they like to go to different organs and different cells in our body. And that's really important for gene therapies, and I'll explain why. They're a really good gene delivery vehicle. They are non-integrating to a certain extent. There's more literature that is coming out now to show that in some cases, or low cases though, in 2 to 4%, that the DNA of the virus does actually integrate into our human genome. It's a very low percentage, but it does actually happen, and it happens randomly as well. They're stable, and they also have long-lasting expression. Okay, here are the different serotypes of AAV. There are 13 of them. These are naturally occurring AAVs. Scientists at the moment are creating novel capsids, which is really exciting because we're quite limited with these AAVs. But there are 13 naturally occurring ones, and they all have subtypes as well. What I want to show you, I know it's a busy slide, but from the 13, you can see, for example, AAV1. It can target the skeletal muscle, and it can target the heart. So if you wanted to develop a gene therapy for a heart condition, you would probably use AAV1 because it really, really likes to go to the heart more than any other AAV. Now, if you focus on where I've highlighted AAV9, AAV9 likes to go to a lot of different organs and different cell types, but it does go to the brain as well. And so AAV9 is used for a lot of gene therapies for the central nervous system. But what the most important thing is about AAV9, as you could see from the previous slide, which I'll just show again, that a lot of the AAVs do like to go to the brain as well. But why we like to use AAV9 is it is one of the AAVs that really, really is good at crossing the blood-brain barrier. Now, normally, you don't want a virus to cross the blood-brain barrier. The blood-brain barrier is this layer of blood cells that connect with our brain. And normally, your blood-brain barrier will prevent any virus from getting into your brain because you don't want it. But in this case, we are taking advantage of AAV9 because it can cross the blood-brain barrier more than any other of the AAVs. And that's why we use it in gene therapies for the CNS. The other thing that I want to highlight is AAV9 is high CNS tropism, which is really important. It crosses the blood-brain barrier. It also has broad tissue distribution. This can actually be to our disadvantage as well because it loves going to the liver. So we have to be really careful about it infecting too many of the other organs and cell types in our body. It also has low prevalence of neutralizing antibodies in the population. This is really important because, as I said, 30 to 60% of us have been infected by AAV, and we don't know because they're non-pathogenic. But when you are infected by a virus, you elicit an immune response, and your body will then remember that you have been infected by a virus. When you have a gene therapy, if your body has already been infected by an AAV, it will elicit a response and will identify that the AAV, it's already seen the AAV in your body, and it will go, no, we've seen you. You're not coming into any of the cells. We're going to stop you. And it will coat the AAV, prevent AAVs from entering the cells. And this is what's so important about understanding whether children have been infected by AAVs, because if you have a high neutralizing antibody titer, then most likely if you do have gene therapy, then you won't be eligible for the trial because it will neutralize the AAV. So there are different mechanisms of delivery, and this depends on the protocol as well. It could be straight IV, intravenous. It could be into the ventricles of the brain. It could be into the spinal cord, or it could be into the back of your brain as well. So there are many different ways in which AVs can be delivered. Okay, how gene therapy works. So we introduce a gene that we want to put into an AAV. This is then packaged into AAV. So as I said, you gut out the viral gene, so they're no longer in the AAV. We then put in our gene of interest, which will be CDKL5. This is then transfused into the body. And what happens is the cell and the AAV are really, really clever. The AAV will attach to the membrane of the cell that it wants to go into, say, for example, with the nucleus. And the cell is very clever. It will allow the AAV to go into the cell, close up the cell again, form a bubble around the AAV, and traffic the AAV all the way through the cell into the nucleus. When it's in the nucleus, what happens is it's really, really clever. The capsid of the AAV will open up a little bit, that purple capsid that you can see, and the green, which is the DNA, will wiggle out of the AAV. And the cell will recognize that this is a human gene, and it'll go, this is human, I know this, I'm going to copy it, and I'm going to make it into a protein. And that's how a normal copy of the CDKL5 gene is made and the normal protein is made. So this is my last slide before I pass on. As you heard earlier, we know that gene therapies are emerging for CDKL5, and we know that they're coming soon. Clinical trials have been carefully designed, and this is really important. The design of them is important because there are going to be eligibility criteria, there are going to be limitations, because it's a trial, it's not a medicine yet. And not all families might be eligible, and one of the reasons would be because of the neutralizing antibodies, but there might be others as well. So I'm going to stop there, and I'm going to pass over to Scott. Do you need this?
It's easier if you're looking. I'm going to build on that talk and try to give you guys a little bit of a sense of like, what does this actually look like? Like, what does it actually mean in a clinic to do a gene therapy? What do you have to go through? This is all theoretical, and it's important to understand some of those concepts, and some of them I'm going to repeat on purpose because they're really important to understand in terms of why we're doing what we're doing clinically. But that was a really wonderful foundation for me to build on and sort of express what does this look like in the real world. So you guys know who I am. I have the pleasure of serving as the Chief Precision Medicine Officer for my institution, and this is my day job, is to figure out how to safely give these types of therapies to patients, not just for neurology, but kind of across our hospital. So what does it really involve? Risk and benefits we're going to talk about for a clinical trial, and a little bit about a clinical trial process and why the clinical trial process happens the way it does, just so that you have that as a framework. This is all obviously leading up to ALAGE. I'm not only going to talk about this for ALAGE purposes, but I'll try to call out where things from this morning's session that Steve mentioned are specific to ALAGE, whereas with a different type of gene therapy program, it may be taking a different approach. All right, we just heard about this. This is another visual, this is repetition, and all this is saying is that up here you have your DNA gets turned into RNA, your RNA gets translated into these chains of amino acids that make proteins. Proteins do functions in your cells. They're the machinery of your cell that helps them work. CDKL5 is the gene. It has to make a protein that helps our neurons stay healthy and function properly and communicate to other brain cells. This is just to mention and Steve mentioned this also, that they're not only working on gene therapies for CDKL5, they are also working on different approaches. So we have ways that we can target things at the DNA level. The ALAJ clinical trial that we're talking about, the RET gene therapy clinical trial is currently happening. There's gene editing that happens. These are all things that happen at the DNA level. You can also target things at the RNA level. You can change the way we process the RNA. That's a processing step in the middle. And sometimes you can help boost the dose or correct something that's an error in processing. Sometimes you can jump right to the end. These are usually called enzyme or protein replacements. And this is, we're going to give the final product, forget the steps at the beginning. The downside of that is you got to keep giving it because our body naturally breaks down proteins that are there so that we can recycle them. So there are examples of this also for other genetic conditions, and that's another type of approach. Now, I'm really just going to focus on the gene therapy part because that's our focus of our session, but I wanted to give you guys some context about why this matters and why we're kind of thinking through these pieces. All right, so if we're targeting the DNA, our goal is to either correct gene editing or replace the gene that's causing disease. Ideally, this is a one and done process. And our biggest concern is around safety right now in many ways. So I say ideally it's a one and done process. The brain puts certain challenges for us, which we're going to get into, which is primarily like, how do you get it into all the brain cells? That's our biggest challenge right now. The change that exists for whatever variant, unless you're mosaic, we have such a sophisticated audience here already asking really good questions. Unless you're mosaic, that is in every cell in the body, it's in every cell in the brain. So you want to get it into every cell in the brain. That's not possible. None of us have the technology to do that right now. So how do we get it into enough cells to make a big difference for patients, but still do it safely? That's the big challenge of brain disorders. The big advantage of brain disorders is that those cells don't replicate. Neurons are the neurons you have after about the age of 2. You're not making any more neurons, so take care of the ones you have, make good choices in life, because you don't get any extra ones. But the advantage of that is because they're not dividing over time, if you can replace it in that cell, it will be used and continue to stay there for, we think, years, maybe forever. And so that's an advantage for neurology if we can get it there. So that makes it ideally a one-time treatment, and it continues to work. But again, how do you get it into the right places, the right organ, the right tissue, the right cells? We talked about replicating or not, and then are there any accidents or off-target things, unintended consequences of trying to do this? And there are issues, and we've already seen some questions around that, around timing of treatment. I think Steve's answer this morning on that was perfect. There's not a window of treatment opportunity that closes. It maybe gets a little smaller. So maybe our impact is a little bit less if you're older, but we don't think right now that the window shuts and you don't have an opportunity for some benefit. This is really reiterating a little bit of what Wendy said already. So the gene will only work if it gets into the right cells. She described this process by which these viruses come, they attach to the outside of the cell, they go into the middle of the cell, which is where the DNA lives inside the nucleus, and they sort of inject the DNA in there, and then you have the DNA ready to go inside your cells, hopefully forever, as long as that cell stays alive and is not replicating. We can give it lots of different ways, and these are some of the important details, and I'll call out sort of the thought around Elijah's approach here. So you can give it IV, like we give lots of other infusions. You can give it into the spinal column through a spinal tap. You can do it surgically, where you're trying to get it into the space around the brain. And sometimes that's into what's called the ventricles, the sort of bags of fluid in the middle of the brain, and sometimes that's into the space around the brain, sort of at the base of the skull or around the neck. Different advantages and disadvantages about these different approaches in terms of how it's going to get to different parts of the brain and the safety associated with it. One of the things that Wendy mentioned a couple of times is about neutralizing antibodies. That means us having antibodies that can actually just block the virus. And that can be a problem, one, from a safety standpoint, if we're going to have a bigger immune reaction, and we're going to talk about immune reactions. But two, it's sort of your own body killing the virus that's supposed to deliver the treatment. So it blocks the treatment that you're hoping for. So that's a real problem. That's the biggest problem when you're giving this IV. when you're giving it directly into the central nervous system, into the fluid around the brain or the spinal cord, that's much less of a problem and not necessarily something that blocks your ability to participate in that treatment or trial. So getting into the risk, I'm gonna go through each of these, and I'm gonna call a little bit of attention to these little pictures we have on the right side of the screen. These you're gonna see again as we go through the workshop, and so I want you to feel like they're familiar a little bit. So there's a couple of different risks that we need to think about when we're doing a gene therapy. A gene therapy, we are doing a gene transplant, and I think that's a useful way to talk about it because in some ways it's similar to doing a bone marrow transplant or an organ transplant. You're putting something that's not already in your body into your body and trying to keep it there for the long run. There are real risks associated with trying to do this that we have to manage very carefully. So you have risk associated with the actual procedure and the infusion itself. Whenever you put something foreign into the body, you can have a reaction to that, like an allergic reaction that can be quite severe in some cases. We talked about this virus likes to go to the liver. When it goes to the liver, it may just cause a little bit of stress for the liver, and the liver is pretty resilient and can bounce back. In some cases, it actually really damages the liver, and you can have liver failure that can be life-threatening. So that's something we have to manage very carefully. It can result in changes in the way your blood clots, and so you can have bleeding or you can have too much clotting, both of which can be damaging to organs and be dangerous. You can have damage to the heart. You can have damage to your nerves and really to your brain. And you can get a really strong immune response. So your immune system's there to help protect you from viruses like the ones we're harnessing to do this work. If your body decides that's a really dangerous virus and it really gets ramped up, sometimes your immune system actually does more damage than the virus does in the first place. And that can happen in certain cases with a gene therapy where the immune reaction actually becomes so severe it can be life-threatening. And then yes, unfortunately, death is a possibility with a gene therapy. We've learned a lot about gene therapies over the last 10 years. As recently as last year, there were patients who died from gene therapy trials. I actually gave a very similar talk to this a year ago for another community. That went forward to a trial, and the first patient who was treated died in that trial. It was a different type of trial, it was a different type of technology, and we have lots of reasons to think that that's not going to happen with Elage. They're using a different approach that's actually been used in other patients already. So we feel more comfortable. That was a different type of capsid. But I say it because I want to be really honest with all of you. I want you to understand the risk and know what we're getting into and come into this with eyes wide open. I get to be the bad guy who gets to scare you all a little bit, and I apologize for that, but it's important. So calling back to these sort of pictures, you can have short-term sort of side effects that may be relatively mild, feeling like you have a flu or something, the steroid side effects, things like that. You can have more severe, requiring hospitalization or even ICU short-term effects, but you get past them and you're okay. Or they have lasting effects, or obviously the worst case scenario, the possibility of death. So how do we avoid all of those things? The biggest thing is controlling the immune reaction that the body is going to have to the virus. And so the mainstay for that is steroids. Steroids kind of block the whole immune system to a certain extent, and we have to use really high doses of this. When we give steroids, we usually are talking about something like 4 to 8 weeks, depending on the exact treatment we're doing. That has issues for your body. There are side effects associated with steroids. You get irritability. That can be real, like real irritability. So for our kiddos, we have to think about that. You can irritate the stomach, which may need another medication to control that. You can have increased vulnerability to other types of infections because your immune system's suppressed. You can have changes in your blood pressure, blood sugar, other things. Again, you might have to use other medications to treat the side effects of the steroids that we have to do for this 1 to 2 months. Sometimes, in addition to steroids, we need other types of immune suppressing medicines. So sometimes it's not just one thing, it's 1 or 2 or 3 treatments that are necessary. When we go directly into the brain, we tend to just do steroids. But sometimes if a person has a reaction like a liver failure, we may have to either increase the dose of steroids, do them longer, or add a different medication. For all those reasons, we have to have really close monitoring. So usually close monitoring for something like a few days to a week, you're in the hospital being monitored constantly. You then get to leave, but you don't get to go real far from the hospital. Usually several times a week, maybe 2 times a week visits, lab draws, closely monitoring the liver, for example, and other things. So you're really staying very close. And that's usually going to be for something like a minimum of 4 weeks, sometimes maybe 6 to 8 weeks. And so depending on how that goes, you can space that out a little bit more. So there's this acute phase in the first few weeks afterwards where we're keeping a really close eye, we're doing lots of visits. It's a whole lot to go through. Again, it's kind of like getting a transplant because we're transplanting genes. And then you get to space out those appointments. If you're not close to the hospital that's doing this, you get to go back home, but you're still going to have to come every few months to every 6 months. Eventually it spaces out probably to once a year, but we need to monitor this long term for long-term side effects as well. And so you're still on the hook for coming in and needing to do that safety monitoring. So all of these things are going to contribute to, again, some of these short-term side effects from the medications we're using to keep you safe, the steroids. And again, there could be more severe short-term side effects in some cases. What about long-term risk? So this is important to emphasize that despite the fact that there actually have been hundreds of patients treated with gene therapies now for various different types of disorders, there is a lot we do not know about gene therapies. When there's something we don't know, it means there's inherently a risk of the unknown. And you have to be honest about that. And if you're doing something in a clinical trial, that's even more true. Because by its very nature, it is a trial. It's not a proven treatment. We don't know how well it's going to work, and we don't know what some of the risks might be that we haven't seen yet in other conditions. We just heard about this. This is supposed to be AAVs don't integrate. So the gene replacement doesn't get into your own DNA, but we actually know sometimes it does, and that means that there is always an increased risk of cancer. That's how we should think about this whenever we're talking about a gene therapy. And actually just a couple of weeks ago, a paper came out where a tumor was seen that was the direct result of a gene therapy that someone got. Again, that's been shown one time concretely out of hundreds of patients treated. So this is a rare scenario. It's not something that we think is going to happen frequently, but it is a risk. By the way, everything we do all day, like going out in front of the sun and all these things, also increase our risk of cancer. But it's something we need to be honest about and talk about, right? So you can have these acute injuries that may actually result in some injury that you don't heal from entirely, and so then you could also have long-term effects from that. So we need to really think through. So again, I'm calling your attention to some of those cards. So realistic expectations, the benefits, the good side, moving past all the bad things. What is a cure? A cure is an intervention that eliminates disease. In the case of CDD and gene therapies, that's not really what we're talking about. We hope we are talking about something that provides meaningful benefit, improvement in quality of life, improvement in functioning. Elimination of disease is not realistic for the vast majority of genetic diseases with current technology. So disease-modifying therapies, we hope, significantly alter the course of disease for the better, but it's not a complete cure. We just can't get enough of these viruses to get into the new DNA into all the cells to actually reverse disease completely. And I want to emphasize this again, I said it already. If we're doing a trial, we don't know all the details about safety and efficacy. It's still research. So we just have to be honest about that. We do have a risk that we cause more harm. We have a risk that there's some injury to nerves, to the brain as part of doing a treatment. One of the things that our data in the ICCRN has shown is that patients who have better functioning also tend to have worse behavior problems. There's a chance that we make patients better and make certain symptoms worse. We're going to have to honestly be prepared for some of those things. We've talked about this a little bit, but if you've already gotten a gene therapy, so far we think that basically means you can't get another gene therapy. People are talking about ways to try to make the science better, the ways that we can block the immune system. But if you've gotten that gene therapy, your immune system's primed to really respond to it. And so we don't think that another dose of another gene therapy would be realistic using the same type of virus. Other people are working on non-virus or other viruses that they could use. Those are more distant into the future, and those might be options. But we need to go into this thinking this is our one-time shot at a gene therapy replacement. Other things, protein replacements, ASOs that target the RNA level that we talked about, those might be options. But for DNA, it's a one-time thing. All right. So the last thing I'm going to say here, you've probably seen something similar to this before. I'm going to just be really brief about this. But what does trial design look like? You've heard a little bit about, like, we do all this stuff in the lab and in animals to try to figure out what's the safest way to deliver this and how do we know what the right dose is and that this works in an animal model. We then move into people. And usually you're talking about something like the first in human, you're going to do this. And that's really just like, can we do it safely? And what's the right dose? And where does it go in the body? And then you start to say, okay, we're going to treat a few patients. And now we're going to look more at safety. And then you go to a phase 2 where you're still mostly looking at safety, but you look a little bit at how well is it working, efficacy. And then you jump into like a main study where your primary thing is, does this work? And also, is it still safe as we treat more patients? And you get more and more patients as you go along this. Phase 4, this one at the end, is usually after the FDA has approved it. You continue to monitor it long term, now being given in as part of clinical care. In the rare disease world, we often truncate this whole thing quite a lot. We just don't have enough patients to try to go through that whole process. And for gene therapies, because they're risky, you want to treat with as few patients as you can to establish safety and to figure out whether it works or not. So we smush these, and sometimes even the phase 3, all together into as few patients as possible where we can really responsibly try to figure out, is this safe and is this working? The safety piece and the small number of patients is because you can imagine if I said, we're going to jump right into treatment and we're going to treat 10 patients at the same time. If something bad happens, you've now exposed 10 patients to that same risk. So a lot of times when we're talking about rare disease, particularly gene therapies, when we're starting the first in human component, we start with one patient at a time, leave a little bit of time to see, make sure everything is okay and safe, and then the next patient, and then the next patient. That little bit of spacing it out is really critical to making sure that it's safe, and we've had time to make sure that it's safe, even though it goes a little bit slower. So that's for safety. It's not to try to restrict access to the gene therapy and to make it so people can't get in. And we start with the smallest number of patients because until we know that it's safe, we don't want to expose more people to a risk than possible. As you get a little further along, you sometimes can start to treat more than one patient at the same time, but in that initial phase, you're trying to be really careful about it. So to summarize, lots of efforts to develop novel treatments for neurogenetic conditions as a whole, and specifically for CDKL5. And we've talked a lot about gene therapy specifically, but there are other approaches as well that are being taken on by Elaj and others. Delivering the gene therapy is like getting a transplant, both short-term and long-term risk, and we have to be really honest about those things so you can weigh the risk-benefit. Gene therapy trials are on the horizon, but these are high-risk, high-reward scenarios, and everyone needs to think really honestly about whether it makes sense for their child. And then I just mentioned this part. We start with really small numbers just to be really safe, and then we increase the number of patients as we show safety and that there is some sort of benefit. And so the number of patients who may have access to treatments is going to increase. But honestly, this is like a several year long process. This is not something that happens in months. And I'll stop with that. And I think we're taking questions at the end, and I'm going to pass it to Peter.
I'm Peter Jacoby. I'm a biostatistician. Tim Benke called me a machine yesterday, which I'm not sure whether I should take that as a compliment or not. As part of the Australian contingent here, I was planning to give you guys a hard time when Australia beat USA in the World Cup, but that didn't happen. That didn't happen. But anyway, as a statistician, I spend most of my working life staring at numbers on a screen, which, well, that wouldn't be for everyone, but I enjoy it. And it's easy for me to forget that behind these numbers are real people and real families who are obviously often struggling to cope with what this condition throws up. So yeah, it's a great opportunity, a privilege really, for me to come here and meet you and be able to talk to the families. So I'm going to talk about the trade-off between risks and benefits, following on from what Scott was talking about. This trade-off occurs in all walks of life, really, whether you're talking about spending money, and it's no different when we're talking about the risks and benefits of a treatment. And my aim is to quantify, put numbers on this trade-off, and using information from a risk-benefit survey, which we plan to administer to as many CDKL5 families as we can. And your inputs from the workshop today will be very helpful in helping us design this survey. How do we quantify this trade-off? I'm going to talk about a technique called the standard gamble, which is a technique widely used by health economists to evaluate states of poor health. Well, I mentioned health economists in an online call recently. Eric thought I said healthy communists, but no, this is nothing to do with communism. So basically, in the context of health economists, the benefit is, if you're talking about somebody in a poor health state, the benefit is a return to perfect health. The cost is the dollars required to achieve this. And what they would be asking is, do the costs justify the potential benefits? And this is the standard gamble. Imagine you are in a state of poor health. And a hypothetical treatment will mean a return to perfect health, but there is a risk, and there is a certain X% risk of death. What value of X would be acceptable for you to go ahead with this treatment? And if it was just a mild condition like headaches or something, then you'd say, no, obviously I'm not going to risk dying because of this. But if it's a serious condition which has affected your quality of life, people might say, yeah, I would be prepared to accept whatever, 25% risk of death maybe. So we're going to sort of develop this technique, and I propose something which I've called a non-standard gamble. And in this survey that I mentioned, we're going to be asking the participants questions like, what risk of a serious illness, and we would have to specify what that serious illness was, what risk of a serious illness would you be prepared to accept in exchange for a large improvement in your child's, for instance, communication? And we would do this. I mean, it would be very hard for somebody to say, 38% or whatever, but we can do this by means of a slider, so people can sort of move it along to give a sort of ballpark estimate, and this works. People have used this before. And of course, then this begs the question, how much improvement is a large improvement? And so we would present scenarios which depend on the current child's ability. For instance, a large improvement might be a child who uses only non-verbal communication, acquires the ability to use spoken words. This would presumably be a large improvement. And again, this workshop that we're going to have just now is going to hopefully help us refine these scenarios. So yeah, the survey will have these questions. What risk of a variety of adverse outcomes of the sort that Scott talked about would you be prepared to accept in exchange for a small or large improvement in in a domain that could be communication or seizures or motor function. And and how will we analyze these responses? Well, we will be able to say, for instance, on average, families would be prepared to accept, say, a 60% risk of kidney failure. I'm not sure that's a good example. I think liver failure, perhaps I should have said. But anyway, in exchange for a large improvement in, say, the child's seizures. And the actual data we get might be like that box plot on the right there. There's a whole huge variety of responses with the mean of 60%. But you know, that in itself, if we see that variety, that's useful information. So that's how we're going to quantify, how we hope to quantify risks and benefits. So why do we want to quantify risks and benefits? Well, statistical modeling, which presumably will be me, will also enable us to rank both the adverse outcomes and the domains for potential improvement in order of the importance that families have told us about. So we might be able to say, for instance, that our risk-benefit data indicate that families regard communication as the most important domain for improvement. These are just examples. We don't know what people are going to say. Or people might even say that the data indicate that, or some families, that death is actually a more acceptable outcome than, say, a loss of functional ability. We don't know. It could be that some families say, look, my child's quality of life is so poor at the moment that I wouldn't accept that getting worse. And results like this have implications beyond gene therapy, really, in just understanding the impact of CDD. So just to summarize, we've got families at the front and center of all this. And the families' perspectives on risks and benefits will inform clinicians in their discussions with families. It will inform industry, pharma. They need to know where to put their money in terms of trials that will offer perhaps the best profile, but best trade-off between risks and benefits. And these results will also inform the whole regulatory landscape, in particular the FDA, who are big on risk benefits. And there's on the right there, that's taken from a FDA document. Different types of patient experience can inform nearly every aspect of the FDA's risk-benefit assessment, including potential benefits that are most meaningful and the acceptability of benefit and risk trade-offs and uncertainty.
Thank you, Peter. All right, so I will talk about a couple of additional consideration, as it were, to the risk-benefit considerations for families. I'll go fairly quickly in the interest of time, and you're more than welcome to address questions afterwards. Come up to me, and I'll be happy to talk. So there's some really special considerations in these early phase gene therapy trials in rare disorders, and when a family makes decisions and weighs benefits and risks, and those are inherent in the informed decision making, the balancing hope with realistic expectations. I actually don't need my glasses. And awareness of short and long-term risks, certainly, but then also financial and opportunity costs, impact on quality of life long-term, and ethical and personal values, right? So a large part of information that families gain about these trials comes from an informed consent process, right? And traditional informed consent includes understanding the medical condition, which in rare disorders is not always complete, reviewing available treatment options, which may be very limited or actually none or just symptomatic care in rare disorders, and then discussing the likelihood of success and potential risks, which are potentially not that well known either. In pediatric disorders, it typically requires some degree of assent, and pediatric assent in our disorders is often limited or not feasible. So families facing serious rare genetic disorders are often in a highly challenging position where treatment options are limited and very complex, right? And meanwhile, they're supposed to make up their decisions about potential benefits, such as meaningful symptom improvement or disease modification, and weighing that against the potential risks. But how can they do that? By gaining information, and very often they have limited access to clear, understandable scientific information. Their primary care provider and other providers that they turn to may be much less familiar in rare disorders with these disease entities and treatment modalities. And there is really quite variable quality information out there from online communities and peer groups. Then there is this concept of prospect of benefit. The FDA actually requires that a pediatric research involving greater than minimal risks, which obviously, as we have heard, is all gene therapy, right, offers the prospect of direct benefit. But in early stage gene therapy research, right, it's research, it may not be possible to really estimate the likelihood of benefit, although we're trying to work towards that. And so speaking to that, in other gene therapy trials, this is from, exit interviews, an ocular gene therapy trial. Families have been asked how they experienced the informed consent process and what it yielded. In principle, I've highlighted it up here. Participants and partners who had received information during the consent process actually had a better understanding of the risks and benefits. However, participants and partners reported still significant deficiencies in communication throughout the trial. So that is a huge hurdle with the information gathering in these very, very complex trials. Then there are challenges as to significant uncertainty regarding the safety, efficacy, durability. There's a perceived potential urgency due to the progressive nature of some of these disorders, and there are few or no alternative treatment options. And families actually tend to perceive these gene therapies with a therapeutic misconception. They believe that this early phase research is actually individualized clinical care, which it is not. And they may overestimate the likelihood of benefit. And that's something to be very, very aware of when you enter these trials, right? Gene therapies are sometimes viewed as a potential miracle cure, and they're not, right? It's very important to keep in mind that responses vary significantly from, at least in our experience, between patients, typically. Some therapies may improve symptoms without curing the disease. Benefits may take quite a while to emerge, so it's not an overnight solution. It's not like taking Tylenol for your headache, right? Or ibuprofen or whatever. And then there's the worry that the effects may or may not be permanent. So families should expect that they will struggle with questions such as, should we participate now? Should we wait for a later trial phase? Should we wait for regulatory approval? Waiting may provide more robust safety data and better defined dosing strategies, but at the same time, your loved one may progress beyond an age of enrollment or disease severity. I will actually skip this slide in the interest of time. Then there is a really heavy burden of unknown long-term risks, right? We've heard about complications that can occur, certainly short-term complications, right? But the long-term risks that I'm talking about is more in the line of the burden on families. The FDA really encourages long-term follow-up in these trials, at least 5 years, ideally 15 years. And this time frame is made to monitor for complications, and families have to come back again and again to the trial sites to monitor for for complications, and that's travel, that's burdensome. They need to coordinate long-term follow-up care, which is complicated. And if there are complications that arise, they need to navigate the management of complications, especially when treatment centers are distant or, for instance, international. Follow-up may not be very easily arranged. Then there are financial and opportunity costs. So pediatric patients obviously require an accompanying caregiver, so a whole entourage, as it were. Adult caregivers must balance research participation with work and family responsibilities. Trials often only have limited sites, and so there is travel and temporary relocation involved. And these factors increase the burden, may add significant financial strain as well. Not all finances can be reimbursed or compensated for. And this is especially taxing for families already burdened by expenses that accompany living with a rare disorder. So maybe to almost finish on here, perspectives from that same paper that I pulled earlier on. One partner of a participant quoted it this way: "That's your whole day. He's missing work, and then I'm taking him there. So we're missing whatever is going on with the other kids. It was a lot. We're lucky because both of our moms live 40, 45 minutes close, so we could work it out like that. If we didn't have those supports in place, and I didn't have anybody to consistently watch my children, I'm not sure how that could have worked out for us." So it really speaks for the complexity of arranging for for support for a very, very complex trial long term. And then finally, there is the quality of life impact, right? There will be separation from other family during prolonged trials, often at a critical stage of a child's life. There is a tremendous, just the other day I was talking to a family, there may be a tremendous feeling of social isolation during a trial because the families can't really communicate everything that's going on with them to other families or their social support groups. And it's a really fundamental factor for them, this feeling of social isolation. And then there's the question, what if there is another sibling with the same condition that does not receive the treatment? I have twins in one trial, and it is certainly quite burdensome for the families to deal with that situation. And then finally, there are some ethical and personal values to take into consideration. Is it acceptable to alter genetic material? Those are really fundamental discussions. What if improvements are only partial? The child gains increasing awareness of her or his own condition without meaningful functional change, right? What does that mean to the quality of life of the child that's being treated? And then what are implications for future generations in some other cases, certainly not ours currently. So they're really deeply personal decisions to be made. Okay, thank you very much.
All right. Well, thank you all for those wonderful presentations. The purpose of everything you've just heard is to give you knowledge about the risks and benefits of gene therapies so we can get your sense of how we weigh that and how we think this is important. As Dr. Jacoby said about this then informs how physicians talk to you, this informs how the companies talk to the regulators, and how people think about this more broadly. So, as a physician, we often talk to parents and families and use words that I'm not sure they fully always understand. And it's something as simple as your doctor says your child has a generalized seizure. I've given talks to many foundations where I say, this is a generalized seizure, and everyone comes to me afterwards and says, no one's ever told me that before, right? So the purpose of Dr. Gold's talk and Dr. Demarest's talk as well is really to give you some foundational information. And these slides will be available for you to look at. And then you can do your own research in this too. We know some of you might have a science background, but most of you don't. So really trying to understand how this is working gives you some foundation to decide what is the risk and benefit of this. And then, you heard a lot about risks through Dr. Demarest and Dr. Suter. And we're not trying to scare you. We're trying to be honest and upfront about really what the risks of this are. We wouldn't be doing this if we didn't think that there's a potential for benefit. And we think there's a real potential for benefit that can outweigh all these risks that you've been told about. But you need to know these risks. And this is the process and trying to come up with a way to quantify how you all think about these risks and benefits, as Dr. Jacoby told you, and then balancing those risks with realistic expectations, as Dr. Suter mentioned. All right, so we have a couple questions for people to ask that we will answer, and then we're going to break out into groups. So who has any questions? There's probably thousands of questions, but we're only going to take two.
So for families like ours, where maybe we're more on the extreme side of developmental outcomes and things like that, or lack thereof, hope is all we have. And the promising studies and technology of gene therapies gives us something to look forward to. But given all the risks and the potential long-term side effects or the unknowns, how do we know it's the right time or not the right time to jump in to something like this? Do we wait for someone who has got maybe more abilities and see what their improvements are before we jump in? Like, it's such an unknown. There's no roadmap, and how do we follow it?
Yeah, so that is part of this discussion, is to hear from you guys and hear what your thoughts are on that and how you weigh the risks that we're talking about with the hope and the potential benefit that occurs. As Dr. Suter said, this is an intensely personal decision you're going to have to make. You're going to have a conversation with your physician, with the research team, with your family, friends, with each other about where you feel this is right for you. And people are, there is no, this is right, this is wrong. There's no right answer to this. You're going to have to decide if it feels right, if you're ready to go forward with it, and it's going to be worthwhile for you. Yes, we have this hope. And one of the risks that wasn't really mentioned, or I think it was mentioned a little bit, is the future, right? Do you do this now, and then what do you have in the future, right? That's a real risk. And the fear of missing out to the future is a real risk. But so you're going to have to balance this all, and it's an impossible task we're asking you all to do. But it's the reality of it, and it's something that you're all going to have to address. And that's why we're having this conversation. This is why we want to hear, as a group, but also individual anecdotes of how you feel about this.
Can I? Thank you so much for such an interesting session. All of you guys, amazing. So I do feel, as mom, that our life is many times choosing between bad and not that bad. And the good option is not on the table. But of course, this gives us tremendous hope. One of the things that you mentioned is that there is risk of having cancer in the future. But long time ago, I heard this interview of this doctor, which specialist was cancer, and he said, back in the days, having cancer was like really bad, no cure. But sometimes nowadays, having cancer is good news in a world of bad news. So my question is, do you know, is there data of what type of cancer our kids could have? Because if it's a very common one with tons of treatment options, that could even be a good news? Or are we talking about a very rare cancer?
Yeah. So the unfortunately, there's now, this was always a theoretical risk that the DNA can integrate into the person's cellular DNA. And if it integrates in a spot that is a cancer predisposition gene, the person could end up with cancer. Theoretical, knew it could happen, but statistically you have billions of base pairs of DNA, it has to insert at the right place. What's the odds of that happening? Now, one case has appeared out of hundreds and hundreds of IV or CNS delivered gene therapy. So we don't know what the risk is. That cancer, it was a unique cancer, but they were able to, according to the paper, do a full resection, a gross total resection. So they think it's, cured from it, but everyone might be a little different because it could integrate into a different part of the DNA. So can't really answer your question. But again, it's the tiny risk of cancer X number of years later versus improving your child for those number of years. Like everyone's going to have to weigh that and how they feel about that. All right, back to you. We're all done.
I will ask you all to join me in thanking Peter, Wendy, Eric, Scott, and Bernard. Thank you.